Okay, so today I'm going to be talking about the multiplication cycles between DNA and RNA viruses, specifically for animal DNA and RNA viruses. I will also be talking about the plus and negative sense and what that all means. So first off, the similarities between RNA and DNA viruses in terms of their multiplication cycles. They both have similar attachment. They both have similar ways of entry, so they go through either fusion or endocytosis. They both do uncoding. They both go through assembly, which is essentially just maturation. And then they both go through some sort of release, so either budding or lysis, depending on whether they're enveloped or not. Same thing with entry, depends on whether they're enveloped or not. The key differences are the site of replication. So DNA viruses replicate within the nucleus of a host cell, whereas RNA viruses replicate within the cytoplasm of a host cell. The next kind of key difference are the type of enzymes that are used. Now, this can be pretty intuitive if you really think about it. DNA uses DNA-dependent DNA polymerase, which they can usually obtain from the host cell. I believe herpes virus is a little bit different. They actually need to encode their own DNA polymerase, but for most DNA viruses, they can just obtain that from the host cell. RNA uses RNA-dependent RNA polymerase. They need to either carry it or produce it. They cannot obtain this from the host, so that's something kind of important to know. Now, the biggest kind of differentiator, or the biggest kind of topic for what I'm going to be talking about right now, is the difference between positive sense and negative sense DNA RNA viruses. Now, when we're looking at, for example, a double-stranded RNA virus, we can see that it has a positive sense strand, and then it also has a negative sense strand. Regardless, because it's double-stranded and it has one negative sense strand, that means it is not ready for translation. It needs to be transcribed into mRNA, or it needs to be converted into mRNA. Now we have a positive sense single-stranded RNA virus. That means it's ready for translation. At that point, a positive sense means it's essentially already mRNA. It can go straight to translation, straight to the ribosomes to get converted into proteins. This is the fastest replication cycle for RNA viruses, because you don't have to get converted into mRNA. It's already mRNA, essentially. You can go straight to the ribosomes. Negative sense RNA are negative sense single-stranded RNA are viruses that are not ready for translation because they are not essentially mRNA yet. They still need to get converted into mRNA. And they do this through the through the RNA-dependent RNA polymerase. Now, a quick reminder, this all happens in the cytoplasm. There are a few exceptions. I believe influenza is an RNA virus that doesn't have to be in the cytoplasm or doesn't replicate in the cytoplasm. I don't know whether or not it actually starts in the cytoplasm, but I know eventually it ends up in the nucleus. So that's something that you can fact check. I'm not sure off the top of my head. The next kind of, or not the next, but for DNA viruses, we don't really care necessarily about the polarity, so we don't care about the negative or the positive sense. It still applies, but when we're actually naming everything, we can just ignore that. Because regardless, it's a DNA virus, so it has to be transcribed into mRNA anyways. So it doesn't matter if it's negative sense, positive sense, it still has to get transcribed in order to go through translation. So we can see that it just goes from, it gets transcribed, it turns into mRNA within the nucleus, then it goes to the ribosomes, and then it goes into translation where it can make all those viral proteins. Now, like I said, DNA viruses usually need to obtain the DNA-dependent DNA polymerase, or I'm sorry, not DNA-dependent DNA polymerase, DNA-dependent RNA polymerase. They will usually need to obtain this DNA-dependent RNA polymerase from the host cell. Overall, DNA viruses need to go through transcription regardless, so we can ignore the polarity in some forms. For RNA viruses, if it is positive sense, it's ready to go straight to translation. We don't have to go through transcription at all. That is essentially already mRNA. Negative sense, single-stranded RNA, they're not ready for translation. They need to go through transcription using that RNA-dependent RNA polymerase. So double-stranded RNA viruses are not ready for translation because they contain a negative sense strand. So they need to go through transcription in order to be turned into mRNA. But yeah, so overall, I hope this was pretty useful, and I hope I made this easy to understand.